Alrighty, I am out this class period, but I will be back on our next class together. So I'm going to leave this video for you to help guide you through what you're supposed to do for the rest of class. We are going to take some notes. We're going to do a practice, and then you're going to do an exit ticket, and you'll turn them all in at the front desk when you're done. Actually, I'll have you turn them in where I usually put the projector on that like white stand-up desk near the front. So the do now that you're going to write in your journal says, how do babies grow in size to become adults? And then number 2 says, what happens to the cells in your hand as you heal from a paper cut? That's after you write the date today. Today is Tuesday, 2/8/22, and the learning objective is students will be able to list the steps of the cell cycle and explain what is occurring in each step. So go ahead and pause this and then answer your do now questions, and then I'll check them when I do our journal check. Okay, so we're going to move on to the notes sheet that you should have picked up on your way in. The notes sheet has some spaces for you to fill out as I go through this video, and it'll be useful to you doing your activities later today. So first off, for the cell cycle in general, living things will grow by increasing the number of cells, not their size of their cells. So your cells can only increase to be so big. They can start out smaller and get bigger, but once they get too big to a certain extent, they get really bad at exchanging things with the outside environment. So instead of continuing to grow in size, they will actually choose to split in half. And that's what we're going to be talking about for this unit. Now, why? Like I showed you, or like I explained, in the beginning, the nucleus itself doesn't change sizes. Only the cell actually, the cytoplasm, the organelles changes sizes. So as the nucleus ratio gets smaller with the cell, it gets harder and harder for that brain or the nucleus to control what's going in and out of the cell because there's so much more space and so much more of the cell to manage. It's like trying to manage a really, really big family versus trying to manage a really, really small family. So the larger a cell gets, the more demands it places on its DNA. And then it has difficulty maintaining homeostasis. So instead of continuing to grow then and making it even harder, they're going to split and make it easier when there's 2 nuclei managing 2 separate volumes. So one cell will become 2 daughter cells when a cell divides. So before it gets too big, it's going to divide, and then one cell will become 2 daughter cells. We use the term daughter and sister a lot in genetics because they're the language that we tend to use for reproductive reasons. And so only really in reality, girls can make more babies or more cells. So we tend to use feminine verbs and feminine nouns. So why does the cell cycle happen? Well, there's 3 major reasons that the cell cycle happens. First of all, we want growth, right? Like I said before, you're not going to actually grow that much in the size of your cell. You're going to grow by growing more cells. The second purpose is to repair. So if you get injured or you get hurt, you're going to repair the area of injury or damage by growing more cells to replace the ones that got damaged or are dead from the incident or the accident. And then the next reason is reproduction. Reproduction is a different kind of cell division. We have actually talked about it before. It's called meiosis. It's not exactly the same as the other two because the other two, for repair and growth, the cells that you're making are identical to replace ones that died, versus for reproduction, you're actually making a cell that is specifically half the normal chromosome count, and then you're also only using it for reproduction. So the cell cycle itself, if you look at this image right here, it's consisting of 2 major parts. One is where the cell is just existing and doing its job, and then the other where it's splitting. And these 2 are both very important to have as part of the cell cycle. So interphase is when the cell is just existing. It is growing, it's developing, it's doing its job. For example, your brain cells are sending electrical signals, your muscle cells are moving, your hair cells are protecting your head and keeping it warm. For example, those things are all things that happen during interphase. And the cell will start from its itty bitty size and grow a little bit bigger during that time too. The other important phase in the cell cycle is called mitosis. And that's the only part of the cell where the cell's actually splitting and becoming 2 daughter cells. And if you look at the diagram right here, you can kind of see that it's not that much of the cell cycle that it happens. It's just like a lot crammed into that short period of time where the cell divides and then it goes back into interphase and continues to do its job as 2 new cells instead of just 1. So inside of interphase, we can break it down. There are 3 parts of interphase. And if you notice right here, this top section right here is mitosis. So we're actually going to ignore that for now. And we're going to talk about interphase, which is 1, 2, 3, as you can see from this inner arrow right here. And the first part of interphase is called the G1 phase. G stands for growth, and 1 stands for the first growth phase. During the G1 phase, cells will grow, they will synthesize or make proteins, and then they will produce RNA. The next step in interphase, still way before mitosis, The middle or next step of interphase is called the S phase. And S stands for synthesis. And synthesis, like I said earlier, means making, right? Here, the cell is going to make more DNA. It's going to replicate or duplicate or double its DNA as it's being synthesized. And you say, well, why would we need to double the DNA? Well, if you're going to make two cells, they both need a copy of DNA. You can just have one copy and split that in half, because then you only have half the instructions or half of your library or half of your information. So we have to duplicate everything so that the new cell you're making also gets DNA. And so that's what happens during S phase, or the synthesis phase. And that's in the middle of interphase. The third part of interphase, right before you actually start dividing, is called G2. And as you guessed it, the G still stands for growth. So it's the second growth phase, and the cells will continue to grow and do the last little bits of prep before it's going to be ready to divide in half and turn into two separate cells. So these three, G1, S, G2, in that order. It's not going to ever go G1, G2, S. It's always going to go G1, S, G2, make up interphase, which is while the cell is existing and doing its job, it's also doing these little things in the background to prepare for when it's about to divide. So why do cells perform DNA synthesis or replication? Well, like I said earlier, it says to make the identical copy of DNA for the new cell that's going to be made in cell division, right? All cells require genetic information, and that's made out of DNA. So the new cell will need those instructions for its purpose during growth, repair, or reproduction. It can't just exist without a DNA in the nucleus. So you have to synthesize that new copy, or copy your DNA inside of that cell to make two versions, so that when you split, one goes to each cell, and then either cell has an entire set of DNA for the instructions to tell it how to grow, how to repair, how to reproduce, if that's its job. DNA replication does occur in the S phase. I know it's kind of weird because S doesn't really stand for anything that sounds like DNA replication, but again, it's because S stands for synthesis, or to make. So DNA replication is the same thing as DNA synthesis. And the S phase, again, is tucked in between the G1 and G2 phases. It goes G1 and then S and then G2 for interphase. And then obviously after G2, you can see in this diagram right here, it goes to the M phase, or the mitosis phase. DNA replication is the process of DNA making a copy of itself. And you can kind of see in this picture, in the beginning, the chromosomes here are actually individual strands, and then they're actually duplicated or replicated or synthesized, and now they're X's. So they have twice the amount, and they're just stuck together in the middle right here. So it kind of looks like a little X like this. And then when they split back up, they'll go back to the original amount, similar to the parent cell. So the two daughter cells are technically identical to each other and to the parent cell. In order to copy your DNA, though, you have to unzip it because it's kind of protected right now. So you have to undo those hydrogen bonds that hold the two backbones and the nitrogenous bases together. So there's an enzyme called helicase. It's the same one that we talked about for transcription. Helicase breaks apart the hydrogen bonds that connects the two strands of DNA like a zipper. It opens it up, and then you're going to have access to all those bases that are now exposed. The template strand is the strand of DNA that you use to make the copy. Use it as a template or a mold or an original form to base the new copy off of. And you'll copy your DNA using DNA polymerase, and that's the enzyme. So as the old DNA unzips, an enzyme or a protein that has a really important job called DNA polymerase brings in new free-floating nucleotides that were in the nucleus. Yes, they're just floating around normally until they're used in this case, and they will be matched up in base pairs A, T, G, C on the template strands as they're built and built and built until the whole DNA strand is copied. They'll also check for errors, because sometimes, like we said, mutations happen when you check for errors and you miss them, or you don't check for errors. But oftentimes, actually, DNA polymerase will proofread its own work. Like you proofread your essay before you turn it in as a final draft, and it will correct any little mistakes that it finds so that there's nothing that's going to be a huge change into the new cell. Then this last part is DNA ligase. An enzyme called DNA ligase then joins together the nucleotides that DNA polymerase brought in. So DNA polymerase did all the building, all the checking, but those pieces are actually still not connected to each other. They're all little pieces that are separate, even though the DNA originally is a long strand. So you got to come in and glue them all together. So ligate actually means to glue or to stick together. And so DNA ligase has the job of sticking all these nucleotides together. And when they're all stuck together, they're new strands of DNA. At the end, replication is complete, and there are two new strands of DNA. But look at the way that they split up. They actually split up. Red goes to one side, blue goes to one side, and then you get that new blue and that new red. So technically, the new double helixes or the new double helices are half old. This is what we call semiconservative. So it says DNA through replication is called semiconservative because half of the new DNA is actually made of the old DNA. This diagram is actually better than the last one. You can see the blue is the old and the yellow is the new parts that are just being built. And the blue is split in half and the yellow is built on top of the blue, but then it is connected to the blue. So once you're done, you can see that the old one was all blue and the new ones are half yellow and half blue. So they're half new, half old. Overall, in a nutshell, here are the steps of DNA replication or DNA synthesis. Helicase will unzip the DNA to open it up for access. DNA polymerase will bring in and build the new nucleotides based on the ATGC pairing rules. Ligase will glue all of the nucleotides together to form the new strands, and then you end up with two identical DNA molecules. And they're semi-conservative because they're half old, half new. Again, overall, when we go through interphase, you have G1, the first growth phase, then you have S, the synthesis phase where DNA is replicated, and then you have G2, the second growth phase before you enter mitosis to actually divide. At that point in G2 though, you should already have double of everything that you need to have double of, including DNA, including organelles, including the ribosomes. All those important things need to already be doubled. Otherwise, you're dividing a cell and the new cell isn't getting enough cell. In your cell cycle though, there are some checkpoints to make sure that when you get to certain points, you're ready to move on to the next step. And there's some important ones because one of them, like in the mitosis part where it tells you, hey, are you even ready to divide or not? So in G1, there's a checkpoint that checks to see if the DNA is ready to be replicated. Sometimes you get to the end of G1 and the DNA is not accessible yet. So you can't actually move into S phase. So then you're paused in interphase. G2 checkpoint checks if the DNA has already been replicated. So obviously this happens in G2 and it's after the S phase. So after you've copied your DNA, your cell actually does a little checkpoint, pauses, and then sees, hey, is the DNA copied? Are there no mistakes? Are we ready to finish growing? Are we ready to divide? And then lastly, there's a third checkpoint in the cell cycle called the M checkpoint, and that's during mitosis, and it checks if these little fibers called spindles are aligning all the chromosomes in the right order for you to then split them apart and go into the respective cells. So these checkpoints are really important because if you get to a checkpoint and you fail to check, then you're not going to continue because that means there's too much of an issue or too much of an error. So you're going to either go back and fix it if it's not a big deal, or if it's a really big deal, the cell actually gets scrapped. It commits apoptosis, known as cell death. And that's only in extreme cases because it's kind of a waste of resources to kill a cell if it just kind of has small things you can fix. So if it's a small issue at the checkpoint, it'll go back and redo all the steps to make sure it's all good. But if it's a really big issue that happened at a checkpoint, then gone. Okay, so overall, this is the cell cycle. Like I said, interphase is this big purple arrow and it represents the majority of the cell cycle. It's like 90% of the time. There's this little arrow over here that actually says G0. Sometimes like your nerve cells, they go into this kind of hibernation where they don't go and divide anymore. That's why we say nerve cells are limited. You don't want to damage your brain cells. You don't want to hurt yourself because they're really hard to regrow because they enter G0. And it's really hard to convince them to go back into the cell cycle to keep dividing. But most cells will just enter G1 right away, go through that checkpoint, go through S phase, go to G2, go through another checkpoint, and then they'll go into mitosis to divide. That being said, if interphase takes up 90% of the time, mitosis only takes about 10% or less of the time. It splits, it's done, moves back into interphase. All right, so for independent practice, there is a sheet at the front that you're gonna pick up, and it is a coloring sheet. You're gonna need color pencils. You're pretty much gonna need one of each color except for like black and white. You're gonna read the instructions on the sheet and follow along. You're gonna be doing a little bit of labeling, a little bit of coloring, and you're gonna turn it in at the front when you're done. And then there's an exit ticket. Make sure you answer the questions and review the success criteria and answer them if need be before you turn it in so that you can get full credit. I will be back in your next class and I will have your stuff graded for you as long as you turn it in to me. You should be able to finish all this. And at any points of this video you were confused or went too fast, make sure you pause, go back and view it so you have your full notes set to help you out with this independent practice part because that's really important for you to review all the info you just gathered from this video. All right, have a good rest of your day. I shall see you next time.